Hello, and welcome to this Microbiology Bio 203 video lecture on microbial genetics. I'm Mr. Kennedy, and I'll be your guide as we explore this topic together. To begin a discussion on microbial genetics, first we must work through some basic terminology. Genetics is the study of genes, how they carry information, how information is expressed, and how genes are replicated. Chromosomes. Chromosomes are structures containing DNA that physically carry hereditary information. Chromosomes contain genes. Genes are segments of DNA that encode functional products, usually proteins. The genome is the sum total of all genetic information in a cell. The genetic code is a set of rules that determines how a nucleotide sequence is converted to an amino acid sequence of a protein. And it is based on the central dogma, which roughly states DNA leads to RNA, which leads to protein. When considering microbial genetics, we must also consider genotypes and phenotypes. The genotype is the genetic makeup of an organism, while the phenotype is the physical expression of genes. DNA and chromosomes. Bacteria usually have a single circular chromosome made of DNA and associated proteins. Microbial DNA can have short tandem repeats, STRs. This is repeating sequences of non-coding DNA. Most DNA transfer in microbes and higher organisms through what is referred to as vertical gene transfer, or the flow of genetic information from one generation to the next. This is an illustration of gene transfer in microbes. DNA replication. DNA replication is the process of making a copy of DNA. DNA forms a double helix whose backbone consists of deoxyribose and phosphate. Two strands of nucleotides are held together by hydrogen bonds between adenine, thymine, guanine, and cytosine, and the strands are antiparallel. The order of the nitrogen-containing bases forms the genetic instructions of the organism. This is a picture of DNA. In DNA replication, one strand serves as a template for the production of a second strand. Topoisomerase and gyrase relax the strands, helicase separates the strands, and a replication fork is created. This is an illustration of DNA replication. DNA polymerase adds nucleotides to the growing DNA strand in the 5' to 3' direction. It's initiated by an RNA primer. The leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously, creating Okazaki fragments. DNA polymerase removes the RNA primers, and the Okazaki fragments are joined by DNA polymerase and DNA ligase. This is an illustration of DNA at the replication fork, working on both the leading and lagging strand, using DNA polymerase. Energy for replication is supplied by nucleotides. A hydrolysis of two phosphate groups on ATP also provides energy. This is an illustration of the energy used for DNA replication. Most bacterial DNA replicate in a bidirectional fashion. Each offspring cell receives one copy of the DNA molecule. Replication is highly accurate due to the proofreading capability of DNA polymerase. This is an illustration of replication of bacterial DNA. RNA and Protein Synthesis. RNA is ribonucleic acid. It is single-stranded nucleotide with a 5-carbon ribose sugar. It contains uracil instead of thymine in its base structure. There are multiple forms of RNA to consider. Ribosomal RNA, or rRNA, is an integral part of a ribosome. Transfer RNA, or tRNA, transports amino acids during protein synthesis. Messenger RNA, or mRNA, carries coded information from DNA to ribosomes. Transcription. Transcription is the synthesis of a complementary mRNA strand from a DNA template. Transcription begins when RNA polymerase binds to the promoter sequence on DNA. Transcription proceeds in the 5' to 3' direction. Only one of the two DNA strands is transcribed. Transcription stops when it reaches the terminator sequence on DNA. This is an illustration of the process of transcription. Translation. The mRNA is translated into the language of proteins. Codons are groups of 3 mRNA nucleotides that code for a particular amino acid. There are 61 sense codons which encode the 20 amino acids. The genetic code involves degeneracy or redundancy, meaning that each amino acid is coded by several codons. This is an illustration of the genetic code in a codon chart. Translation of mRNA begins at the start codon, AUG. Translation ends at the nonsense codons, UAA, UAG, or UGA. Codons of mRNA are read sequentially. tRNA molecules transport the required amino acids to the ribosome. tRNA molecules also have an anticodon that base pairs with the codon. Amino acids are joined by peptide bonds. This is an illustration of the process of translation as it begins at the start codon. Here, a second tRNA is added, and the amino acids of the two tRNAs join. Here, the first tRNA is released, and the ribosome moves on to the next set of codons, repeating the process over and over until a stop codon is reached. This is an illustration of what occurs at the stop codon. The ribosome will release the mRNA strand and a sequence of amino acids made by reading the mRNA strand. In bacteria, translation can begin before transcription is complete. This is an illustration of the simultaneous transcription and translation that can often occur in bacterial cells. Transcription in eukaryotes. In eukaryotes, transcription occurs in the nucleus, whereas translation occurs in the cytoplasm. Exons are regions of DNA that code for proteins, while introns are regions of DNA that do not code for proteins. Small nuclear ribonucleoproteins remove introns and splice together exons. This is a picture of RNA processing in eukaryotic cells. Regulation of bacterial gene expression. Constitutive genes are expressed at a fixed rate. Other genes are expressed only as needed and are referred to as inducible genes and repressible genes. They can be controlled through catabolite repression. Repression inhibits gene expression and decreases enzyme synthesis. It's mediated by repressors, proteins that block transcription. Their default position of a repressible gene is on. Induction turns on gene expression. It's initiated by an inducer. The default position of an inducible gene is off. The operon. The operon is made up of a promoter segment of DNA where RNA polymerase initiates the transcription of structural genes. The operator is the segment of DNA that controls the transcription of structural genes. The operon is a set of operator and promoter sites and the structural genes they control. In an inducible operon, structural genes are not transcribed unless an inducer is present. In the absence of lactose, the repressor binds to the operator, preventing transcription. In the presence of lactose inducer binds to the repressor. The repressor cannot bind to the operator, and transcription occurs. This is an illustration of an inducible operon. This is an illustration of repressor active, operon off. Operons. The repressor protein binds with the operator, preventing transcription from the operon. This is an illustration of repressor inactive, operon on. When the inducer, allolactase, binds to the repressor protein, the inactivated repressor can no longer block transcription. Structural genes are transcribed, ultimately resulting in the production of the enzymes needed for lactose catabolism. Repressible operons are structural genes that are transcribed until they are turned off. Excess tryptophan is a co-repressor that binds and activates the repressor to bind to the operator, stopping tryptophan synthesis. This is an illustration of the structure of the operon. The operon consists of the promoter, P, operator, O sites, and the structural genes that code for the protein. The operon is regulated by the product of the regulatory gene. This is an illustration of the repressor inactive, operon on. The repressor is inactive and transcription and translation proceed, leading to the synthesis of tryptophan. This is an illustration of the repressor active, operon off. When the co-repressor, tryptophan, binds to the repressor protein, the active repressor binds with the operator, preventing transcription from the operon. Positive regulation. Catabolite repression inhibits cells from using carbon sources other than glucose. Cyclic AMP can build up in a cell when glucose is not available. Cyclic AMP binds to the lac promoter, initiating transcription and allowing the cell to use lactose. This is an illustration of the positive regulation of the lac operon. Epigenetic control. Methylating nucleotides turns genes off. Methylated off genes can be passed to offspring cells and are not permanent. Post-transcriptional control. MicroRNAs base pair with mRNA to make it double-stranded. Double-stranded RNA is enzymatically destroyed, preventing the production of a protein. This is an illustration of microRNAs control and how they control a wide range of activities in cells. Changes in the genetic material. Changes in the genetic material are often referred to as mutations. Mutation is a permanent change in the base sequence of DNA. Mutations may be neutral, beneficial, or harmful. Mutagens are agents that cause mutation. Some mutations are spontaneous mutations which occur in the absence of a mutagen. Base substitutions or point mutations are the change in one base in DNA. This is an illustration of a base substitution. Missense mutations are base substitutions that result in a change in an amino acid. This is an illustration of a type of mutation and their effect on the amino acid sequence. Nonsense mutations are base substitutions that result in a nonsense or stop codon. This is an illustration of, again, a type of mutation and its effect on the protein sequences. Frameshift mutation is an insertion or deletion of one or more nucleotide pairs. This shifts the translational reading frame. This is an illustration of a frameshift mutation and its effect on the protein sequence. Chemical mutagens. Nitric acid is a chemical mutagen which causes adenine to bind with cytosine instead of thymine. Nucleoside analogs can be incorporated into DNA in place of a normal base and cause mistakes in base pairing. This is an illustration of the oxidation of nucleotides, which makes a mutagen. This is an illustration of the altered adenine pairings with cytosine instead of thymine. This is an illustration of the nucleoside analogs and the nitrogenous bases they replace. Radiation. Ionizing radiation, such as X-rays and gamma rays, causes the formation of ions that can oxidize nucleotides and break the deoxyribose phosphate backbone. UV radiation causes thymine dimers. Photolyases separate thymine dimers. Nucleotide excision repair can also cut out incorrect bases and fill in the correct bases. This is an illustration of the aforementioned processes. The frequency of mutation. Spontaneous mutation rates are roughly 1 in every 10 to the 9th replicated base pairs, or 1 in 10 to the 6th replicated genes. Mutagens increase the mutation rate to 10 to the -5 or 10 to the -3 per replicated genes. Identifying mutants. Identifying mutants can be done directly or indirectly. Positive or direct selection detects mutant cells because they grow or appear different than mutated cells. Negative, indirect selection detects mutant cells that cannot grow or perform a certain function. Auxotrophs are mutants that has a nutritional requirement absent in the parent. The Ames test exposes mutant bacteria to mutagenic substances to measure the rate of reversal of the mutation. It indicates the degree to which a substance is mutagenic. Genetic transfer and recombination. Genetic recombination is the exchange of genes between two DNA molecules, which creates genetic diversity. Crossing over is a natural form of genetic recombination where two chromosomes break and rejoin, resulting in the insertion of foreign DNA into the chromosome. This is an illustration of genetic recombination by crossing over. Vertical gene transfer is the transfer of genes from an organism to its offspring. Horizontal gene transfer is the transfer of genes between cells in the same generation. Transformation. Transformation occurs when genes are transferred from one bacterium to another as naked DNA. Transformation was first seen by Griffith's experiments on pneumonia-causing bacteria in the early 1900s. The mechanism of genetic transformation in bacteria is illustrated here. Conjugation. Conjugation is a form of sexual reproduction in bacteria. Plasmids are transferred from one bacterium to another. It requires cell-to-cell contact via a sex pilus. This is an illustration of bacterial conjugation. In bacterial conjugation, there are no male or female cells. Donor cells carry the plasmid F factor and are called F+ cells. HFR cells contain the F factor on the chromosome. This is an example of conjugation in E. coli. Conjugation can be used to map the location of genes on a chromosome. Transduction in bacteria. DNA is transferred from a donor cell to a recipient via bacteriophage. Transduction can occur in either generalized or specialized forms. Generalized transduction occurs when random bacterial DNA is packaged inside a phage and transferred to a recipient cells. Specialized transduction occurs when specific bacterial genes are packaged inside a phage and transferred to a recipient cell. This is an illustration of transduction by bacterium. Plasmids. Plasmids are self-replicating circular pieces of DNA, 1 to 5% the size of bacterial chromosomes. They often code for proteins that enhance the pathogenicity of a bacterium. This is an illustration of R factor, a type of plasmid found in bacteria. Plasmids can be conjugative plasmids, which carry genes for sex pili and transfer of the plasmid. Dissimilation plasmids encode for enzymes for the catabolism of unusual compounds. Resistance factors, R factors, can encode antibiotic resistance. Transposons are segments of DNA that can move from one region of DNA to another. They contain insertion sequences, or IS sequences, that code for transponase that cuts and reseals DNA. Complex transposons can also carry other genes, as an example, antibiotic resistance. Genes and evolution. Mutations and recombination can create cell diversity. Diversity is the raw material for evolution, and natural selection acts on populations of organisms to ensure the survival of organisms fit for a particular environment. This concludes our coverage of microbial genetics.